Hello everybody, I am Somukantigiri. Welcome to my channel. So in this video we will discuss in detail about the immunology. Okay, so the entire chapter of the immunology will be discussed in this video. So let's start. So see the contents. Okay, what is immunity, it will be discussed first. After that we will discuss about the human immune system. Okay, then types of immunity will be discussed. Then we will discuss about the differentiation between your innate immunity and adaptive immunity. Okay, then we will discuss about immune responses, different types of immune responses, primary immune responses and secondary immune responses. They will be discussed after that. Then we will discuss about the lymphocytes, T lymphocytes and B lymphocytes in detail, their role, their your classification, their types. Okay, all will be discussed in this video. Okay, so after that we will discuss about the antigen. Okay, antigen means the epitope of antigen, paratope of antibody. Okay, hapten, all will be discussed. Then we will discuss about antibody, the structure of antibody, enzymatic digestion of antibody and types of antibody and different salient features of different antibodies, that will be also discussed. Okay, then we will briefly discuss about the antigen antibody reactions. We will also discuss about affinity, avidity and their differences. Okay, then after that we will briefly discuss about the complement system and major histocompatibility complexes. So let's start. This is my brief introduction. So let's start. What is immunity? Okay, so immunity means the resistance offered by the host against the microorganism. That means when a microorganism is going to invade inside our body's immune system got activated. After that, the immune system, our immune system actually provide immunity against the microorganisms, invading microorganisms. Okay, so that is immunity. So immunity is defined as the resistance offered by the host against the microorganism or any foreign substances. Okay, so the foreign substances which are going to enter inside our body, they are known as antigen. Okay, they are known as antigen. So what is immunology? Immunology is a branch of science, or you can say immunology is a branch of life science or medicine, whatever may be, that covers the study of immune system. Okay, that actually deals with the study of our immune system, that is your immunology. And all of you know that the father of immunology is Edward Jenner. Okay, Edward Jenner is regarded as the father of immunology. Okay, so what is antibody? So what is antigen? We have discussed. Antigen is the foreign invading molecule that is going to enter inside our body. That is antigen. And antibody is the response of our body's immune system. Actually, they are specialized components of our immune system that is going to kill the antigens. Okay, so they are your antibody. Okay, so coming to the next point, human immune system. See, the human immune system has three different components, like tissue cells, you know, the phagocytes, you know, the macrophages. Okay, you know, the Kupffer cells, they are the your tissue cells. Okay, so after that, soluble molecules are there. Antibodies are coming under in this class, soluble molecules. Then lymphoid organs are there. In case of lymphoid organs, examples are thymus, bone marrow, tonsil, lymph, and you can see the position of that lymphoid organs. Okay, but the lymphoid organs are further classified into two categories. One is your primary lymphoid organ and another is your secondary lymphoid organ. Okay, so what are the primary lymphoid organs? They are the organs where B and T lymphocytes mature. Examples, thymus and bone marrow. Okay, that is very, very important. And what is your secondary lymphoid organ? Secondary lymphoid organ means here the lymphocytes react with antigen. That means antigens are going to enter inside our body, that the invading microorganisms or invading molecules, and after that they are going to react with lymphocytes in secondary lymphoid organs. Examples are spleen, lymph nodes, tonsils, etc. Okay, so coming to the very, very important topic, types of immunity. Okay, in case of different examination, you expect 1 to 2 questions from this particular point, types of immunity. It is very complex, but simultaneously it is very easy to understand. Okay, just try to learn these points. See, immunity can be divided into two types. One is innate immunity and another is adaptive immunity. Innate immunity means the immunity provided to you by birth. Okay, that is by birth. Okay, you are getting this by birth. Okay, adaptive immunity means this is acquired immunity. Okay, you are acquiring experiences throughout your life. Okay, and after that your immune responses are getting converted according to those experiences. Okay, so that is your adaptive immunity. Adaptive immunity further can be classified into two categories. One is active and another is passive. Active means here the antigens are going to enter inside your body, that is active. And in case of passive, passive means antibodies are going to enter or developed or given to your body. Okay, that means see देखो जो adaptive immunity होता है ना, that can be further classified into two categories. One is active and another is your passive. Active means by antigen and passive means by antibody. What is the meaning of this? देखो active can be further divided into two categories. One is natural and another is your artificial. So natural active immunity is there and artificial active immunity is there. That means this antigen is going to enter inside your body through the natural processes or maybe artificially given by vaccination. So the natural active immunity means examples are infections, different types of infections. Okay, in case of different types of infections, antigens are going to enter inside your body through the natural processes, and after that your body is going to develop adaptive or acquired immunity. So that's why natural active adaptive immunity. Okay, got your point. If the antigen is going to enter inside your body by artificial methods like vaccination, you know the BCG vaccine. So the BCG vaccine is a life attenuated vaccine where the microorganism is attenuated. That means its potential, its immunogenicity has been lower. Okay, so that's why it is going to enter in, it is given to your body to produce adaptive immunity. Okay, so I hope this is clear. Active immunity is clear. Okay, and then coming to the passive immunity. What is passive immunity? See, passive immunity means antibody is given to your body. Okay, antibody is given to your body, and again the antibody can go into your body through natural processes or artificial processes. Okay, so example of natural processes is your mother to fetus IgG transfer. When a baby actually situated inside the mother's womb, then the IgG जो antibody होता है ना, that actually go into the fetus and protect the newborn. Okay, so that is the natural passive immunity. In case of artificial passive immunity, what happens? See here, if one is there, like he was working in the field and suddenly a cobra snake bites him, so you will suddenly take the person to the hospital. Okay, so वहाँ पे क्या होगा, वहाँ पे आपको एक एंटी स्नेक एंटी वेनम दिया जाएगा. ठीक है, so that particular snake antivenom is the proper particular specific antibody against the snake venom, against the snake's antigen. So that is your artificial passive immunity. Okay, so this slide is very very important. You have to understand the whole classification of immunity with example very well, क्योंकि it is frequently asked in different exams. Okay, so then coming to the difference between innate immunity and adaptive immunity. What are the difference? See, the first difference is innate immunity is by birth. Okay, whereas the adaptive immunity is the during your lifetime. Okay, during your lifetime you will experience several antigens and your body's immune system will be developed after that. Okay, here देखो, in case of innate immunity, immune responses occurs in minutes. Okay, and in case of adaptive immunity, it's a long process. It may take several days. Okay, so in case of innate immunity, prior exposure to the antigen is not required. This is very important. Whereas in case of adaptive immunity, prior exposure to the antigen is the main criteria. Okay, in case of innate immunity, memory response is absent. That is very very important. And in case of adaptive immunity, your cell, your immune system will store a memory, a replica of the invading antigen. Okay, सो फर्दर जो अटैक होगा ना बाद में वो रुका जाएगा. तो दैट्स वाई the immunological memory responses are present in case of adaptive immunity, whereas it is absent in case of innate immunity. Okay, and the last point is diversity is limited. Okay, so diversity in case of innate immunity is limited, whereas in case of adaptive immunity wide diversity is present. Okay, so coming to the components of innate immunities. See, the components of innate immunities are very obvious. Anatomical barriers like skin, okay, mucosa, they are the first line barrier of our body. Okay, then physiological barriers like body temperature, different cells like phagocytes, neutrophils. Okay, they are the components of innate immunity. Whereas in case of adaptive immunity, you will find two main components. One is your T cell, that means T lymphocyte, and another is your B lymphocyte. We will discuss in detail about the T lymphocyte and B lymphocyte in the later part of this video. Okay, so then coming to the immune responses. What are the immune responses? Immune responses means antigen is going to enter inside your body. Okay, and your body will, actually your body's immune system will generate a response against the antigen. Okay, so the responses are classified into two categories. One is primary immune response and another is your secondary immune response. Primary immune response means when a particular antigen, let's say A, that is going to invade to your body for the very first time, so your body's immune response will be termed as your primary immune response against A. Okay, so secondary immune response, that means secondary immune response means when A is going to attack subsequently, मतलब फास्ट अटैक उनका हो गया है, उसके बाद सेकेंड टाइम वो आ रहा है अटैक करने के लिए उस आपका बॉडी को. ठीक है, सो in that particular situation your body's immune response towards the antigen A is regarded as your secondary immune response. Okay, so secondary immune response is the body's reaction to the subsequent attack of the same microbes. Okay, so that is your secondary immune response, and primary immune response is body's reaction to the first attack of microbes. Okay, so keep the point in mind, in your mind. Okay, and all these immune responses are carried out by your B lymphocyte and T lymphocyte. So let's understand what are the B lymphocytes and T lymphocytes, their roles, their functions, and their classifications. Okay, that is very very important. देखो, lymphocytes are the specialized WBC cells. Okay, they can be classified into two categories. One is T lymphocyte and another is your B lymphocyte, and again one class is there, that is your NK cell or natural killer cells. So they can be classified in three types: T lymphocytes, B lymphocytes, and natural killer cells. Okay, so T lymphocytes can be further classified into helper T cells, that is known as CD4 plus cells, cytotoxic T cells, CD8 plus cells. Okay, and B lymphocytes can be classified into two categories. One is plasma B cells and another is your memory B cell. Okay, so coming to the differences between the T lymphocyte and B lymphocyte. What are the differences? देखो, the main difference is the site of maturation. Actually, T lymphocyte and B lymphocyte, they both originates from your bone marrow. Okay, but their site of maturation is different, because T lymphocyte it actually matures in your thymus. Okay, whereas B cell matures in bone marrow. That's why B for bone, your T for thymus. Okay, that's why the name came. And one more important thing is that receptors on the cell surface. Okay, so only the B lymphocyte cells have receptors on its surface, whereas no T cell has such receptors. Okay, so these are the differences between T lymphocytes and B lymphocytes. Okay, so coming to the critical analysis, coming to the roles or functions of different T cells and B cells. What are their roles? Okay, so first your helper T cell, that is also known as CD4 plus T cells. Okay, देखो, immunity can be also classified into two categories. Okay, depending on the antibodies involvement. Okay, so humoral or antibody mediated immune response one is, and another is your cell mediated immune response or CMI. Okay, so AMI, that is your antibody mediated immune response, and another is your CMI, cell mediated immune response. So these two types of immune responses are also there. Okay, so in case of AMI or antibody mediated immune response, in that particular cases antibodies are involved to produce immune response, but In case of cell-mediated immune response, no antibodies are involved, whereas some cells, specialized cells like your macrophages, phagocytes, okay, cytotoxic cells, okay, cytotoxic T lymphocyte cells, they actually engulf the invading microbes, okay, and eliminate them from the circulation, okay. So these two types of immune responses are there, and helper T cell actually helps in these two types of immune responses, okay. These constitute 50 to 70% of the all T cells and usually designate as the Th cells, okay. These cells help B cells and other T cells to multiply into large clones and carry out their role in the immune response, okay. And more specifically, there is a common pathway, okay, before your AMI and CMI. That means before your antibody mediated immune response and your cell mediated immune response, there is a common pathway, okay. That pathway involves the presentation of antigen to the helper T cell, okay. उसके बाद क्या होता है, helper T cell एक्टिवेट हो जाता है, ठीक है. And after that helper T cell differentiates into two forms, one is Th1 and another is your Th2. If Th1 produce, then CMI, I mean cell mediated immune response will be generated. If Th2 is produced, then your antibodies will be produced. After that, antibodies will be produced and your antibody mediated immune responses or AMI will be generated. So helper T cells plays a critical role in your immune system. When antigen is going to enter inside your body, first that is going to be presented before the helper T cell. उसके बाद helper T cell एक्टिवेट होगा, उसके बाद helper T cell का डिफरेंशिएशन होगा. दो टाइप है, Th1 है, Th2 है, ठीक है. If Th1 is generated, then your cell mediated immune response will be generated. उसके बाद, ठीक है. If Th2 is produced, then your antibodies will be produced after that, and your antibody mediated immune responses will be generated. So these are the critical functions of your helper T cell. Then coming to the cytotoxic T cell, that is known as CD8 plus cells, okay. They actually secrete cytotoxic, that means some toxins will be secreted from those cells, and they are known as your lymphotoxins, okay. They has the ability to kill the virus infected cells. That's why they are cytotoxic in nature. And in case of B cells are divided into two categories. One is plasma B cell and another is your memory B cell, okay. So plasma B cells are actually known as the effector cells and they produce the antibodies. That's why they are also known as the antibody factory inside your body, ओके. सो जो एंटीबॉडीज आपका जो प्रोड्यूस हो रहा है ना बॉडी में, तो वो प्लाज्मा बी सेल्स ही कर रहा है आपका बॉडी में, ठीक है. उसके बाद मेमोरी बी सेल करता क्या है, देखो मेमोरी जब आपका एक एंटीजन आएगा ना, मान लो ये एंटीजन है, दैट इज गोइंग टू इन्वेड इन योर बॉडी, ओके. तो वहाँ पे बॉडी का जो इम्यून सिस्टम है ना, उसका एक रेप्लिका स्टोर करता है, क्योंकि सब्सिक्वेंट अटैक उसका रोका जाए उस एंटीजन का, ठीक है. सो मेमोरी जो स्टोर हो रहा है ना, वो कौन सा सेल स्टोर कर रहा है, मेमोरी बी सेल है, वो स्टोर कर रहा है, ठीक है. मेमोरी का जो एंटीजन आ रहा है ना, उसका मेमोरी स्टोर कर रहा है मेमोरी बी सेल्स. सो द सब्सिक्वेंट अटैक ऑफ दैट पर्टिकुलर एंटीजन कैन बी प्रिवेंटेड, okay. So reinfection can be prevented. That's the function of your memory B cell. Then coming to the natural killer cell. The natural killer cell is cytotoxic in nature, not antigen specific. They are not antigen specific, okay, and they are a part of innate immunity, okay. So coming to the antigen, देखो एंटीजन इज वेरी इंपॉर्टेंट. उसके बाद एंटीबॉडी हम लोग पढ़ेंगे, वो भी बहुत अच्छे से पढ़नी चाहिए, ठीक है. सो एंटीजन होता क्या है, देखो एंटीजन है एक इन्वेडिंग मॉलिक्यूल जो बाहर से आ रहा है, एक फॉरेन पार्टिकल, फॉरेन माइक्रोब्स जो बाहर से आ रहा है बॉडी के अंदर में, ठीक है. उसके बाद क्या होगा, उसके बाद बॉडी का इम्यून सिस्टम जनरेट होगा, इम्यून सिस्टम आपका एक्टिवेट होगा. बट ऑल एंटीजेंस कैन नॉट प्रोड्यूस योर इम्यून रिस्पॉन्सेस, क्योंकि जो एंटीजन आपका आ रहा है ना बाहर से, उसका कोई सर्टेन क्राइटेरिया होनी चाहिए. सो दैट इज द टर्मिनोलॉजी एंटीजेनिसिटी. What is antigenicity? It is the ability of an antigen to induce an immunological response. So all antigens cannot produce your immunogenic responses. One example is there, that is hapten. Okay, hapten is a low molecular weight compound. Hapten is a low molecular weight compound. Okay, that is antigen, but that does not have antigenicity. That means hapten cannot produce your immune responses. When hapten is binded to adjuvant, then the entire compound hapten plus your adjuvant, that can uh, activate your immune responses. Okay, so the difference I think is clear to you. So antigenicity, what is antigenicity? It is the ability of an antigen to induce an immunological response when it is encountered by the human body. Okay, and antigenicity can be of two types. One is your immunogenicity, okay, and another is your allergenicity. That means immunogenicity means your antigen is going to invade in your body, that is okay, but उसके बाद जो आपका इम्यून रिस्पॉन्स जो जनरेट होगा, सो वो नॉर्मल है कि एबनॉर्मल है. जो ability of antigen to induce normal immune responses in the body, that is your immunogenicity. Okay, and allergenicity, what is allergenicity? That means it refers to the ability of an antigen to induce abnormal immune responses. That means your antigen is there, that is going to enter inside your body. If the antigen is your antigenic in nature, then it is going to activate your body's immune system. So the immune response can be of two types. One is your normal immune response and another is your abnormal immune response. If the normal immune response is generated, then that is known as immunogenicity of your antigen. If the antigen is going to enter inside your body to produce normal immune responses, then it is known as immunogenicity. If it is going to produce abnormal immune responses, then it is known as allergenicity. Okay, I hope this is very clear to you. Then coming to epitope and paratope. See, what is epitope? Epitope is a part of your antigen. Okay, it is also known as your antigen determinants. Okay, it's the smallest part of your antigen that is going to produce your immunogenic responses. What is paratope? Paratope is a specific site of antibody. Paratope is a specific site of antibody that reacts with the antigen's epitope. जो एंटीजन एंटीबॉडी का जो रिएक्शन होगा ना बॉडी के अंदर में, वो एक्चुअली एपिटोप एंड पैराटोप का बीच में होगा. ठीक है. सो हैप्टेंस, देखो हैप्टेंस आर द लो मॉलिक्यूलर वेट मॉलिक्यूल्स, आई हैव ऑलरेडी टोल्ड यू दैट, एंड दे हैव लैक ऑफ इम्यूनोजेनिसिटी. दैट मीन्स कैन नॉट इंड्यूस इम्यून रेस्पॉन्सेस बट can bind to their specific antibody. When hapten becomes immunogenic, now when it is combined with a larger protein molecule that is called as carrier or adjuvant. See, hapten is an antigen but does not produce immune response inside your body. Okay, when hapten binds to carriers or adjuvant, then the entire molecule actually going to enter inside your body and can produce immunogenic responses against it. Okay, then coming to the point, factors influencing immunogenicity. What are the factors influencing immunogenicity? See, the size of antigen, it is very important. If the size of antigen is bigger, then immunogenic response will be more. Okay, chemical nature. See, the proteins are stronger immunogens than carbohydrates. Okay, that is the point. Then structural complexity. See, the structural complexity means the structural complexity and your immunogenic responses, those are proportionally related. Okay, that means structural complexity of the antigen increases the immunogenicity. Okay, and adjuvant, if the adjuvant is added to some antigen, then more immunogenic responses will be created. So that's why vaccines, in case of vaccines preparation, so adjuvants are used to bind with antigens to produce more immunogenic responses. Then coming to the antibody. See, first let's study about the structure of your antibody. Okay, so and this is your antibody. Actually, 2 light chains and 2 heavy chains are there. Okay, in case of antibodies, 2 light chains and 2 heavy chains are there. This is your heavy chain, this is your heavy chain, this is your light chain, and this is your light chain. Okay, and some bonds are there like sulfide disulfide bonds, okay, and some salt bridge are also there, salt linkages are there, hydrogen bonds are there, hydrophobic bonds are there. Those are responsible for holding the structure, holding the integrity of the structure of your antibody. Okay, so antibody are the Y-shaped molecule composed of 4 polypeptide chains, 2 light chains and 2 heavy chains. Why they are known as light chain? Because the molecular weight is low. Why they are called as heavy chains? Because their molecular weight is high. That's why. Okay, and the chains are held together due to different bonds like disulfide bonds, hydrogen bonds, hydrophobic bonds, salt linkage, etc. Okay, and one more important thing, the light chain and heavy chains can be further subdivided into two categories. One is variable region and another is your constant region. Variable region actually responsible for antigen binding. See, this is your antibody. Okay, this is your heavy chain, this is your heavy chain, this is your light chain, this is your light chain. Okay, so देखो, each chain has two components. One is variable region, V is denoting that, and C constant region. Actually, antigen binds to the variable chains. वेरिएबल जो पोर्शन है ना चेन का, वहाँ पे एंटीजन आकर बाइंड होगा. सो द एपिटोप जो हमने पढ़ रखा ना, जो एंटीजन का मिनिमम, एंटीजन का स्मॉलेस्ट यूनिट है, वो आकर यहाँ पे बाइंड होगा. ठीक है, वेरिएबल पोर्शन ऑफ योर एंटीबॉडी वहाँ पे बाइंड होगा. और जो आपका जो एंटीबॉडी का पैराटोप है ना, वो कहाँ पे है? वो एक्चुअली आपका जो वेरिएबल पोर्शन है ना, वहाँ पे ही है आपका एंटीबॉडी का पैराटोप. ओके, सो वन मोर थिंग इज दैट, देखो दिस इज अ वाई शेप मॉलिक्यूल. जो वाई शेप का जो नीचे वाला पोर्शन है ना, उसको हम लोग एफ सी बोलते हैं. ठीक है, और जो ऊपर वाला पोर्शन है ना, उसको हम ए बी बोलते हैं. एफ ए बी मीन्स फ्रैगमेंट वेयर एंटीजन बाइंड्स. ओके, एंड एफ सी मीन्स फ्रैगमेंट दैट इज क्रिस्टलाइजेबल. ओके, so from those particular points the name came. Okay, so one more thing is that your, in case of antibody structure, that is your hinge region. That is very very important. देखो hinge region, देखो आपका जो यहाँ पे देखो आपका जो ये वाला जो चेन है ना, इसको हम बोलते हैं हेवी चेन. ये हेवी चेन जो है ना, लाइट चेन हेवी चेन, ये हेवी चेन है. यहाँ पे देखो दो कांस्टेंट रीजन है. ठीक है, दो कांस्टेंट रीजन का बीच में एक गैप है, एक ग्रूव है, ग्रूव. ठीक है, उसको हम लोग हिंज रीजन बोलते हैं. यहाँ पे होता क्या है, देखो प्रोलीन और सिस्टीन, ये जो दोनों एमाइनो एसिड है ना, यहाँ पे बहुत सारे मात्रा में अवेलेबल है. एंड वन मोर थिंग, ऑल द एंटीबॉडीज डू नॉट हैव हिंज रीजन, ओनली GAT, GAD, that means IgG, IgA and IgD, they have the hinge region. Okay, IgE, IgM do not have hinge region. आपको एग्जाम में पूछा जा सकता है. ठीक है, देन कमिंग टू द एंजाइमेटिक डाइजेशन ऑफ एंटीबॉडी. आपको जो एंटीबॉडी है उसको तोड़ना है, उसका स्ट्रक्चर आपको तोड़ना है. तो कैसे तोड़ेंगे आप? सो तीनों तरीके, मतलब 3 डाइजेशन स्टेप्स आर देयर. ओके, वन इज पैपेन, another is pepsin and another is your marketoethanol digestion. Okay, so in case of papin digestion, papin cleaves the Ig molecule, that means immunoglobulin or antibody molecule, at a point above the hinge region. Okay, just above the hinge region or at the hinge region. Okay, क्वेश्चन आपको पूछा जा सकता है कि पैपिन कहाँ पे ब्रेक करेगा, एंटीबॉडी का जो स्ट्रक्चर है कहाँ पे ब्रेक करेगा. हिंज रीजन के बहुत से, मतलब हिंज रीजन के ऊपर में, नीचे में और कहीं भी कर सकता है. ठीक है, so papin digestion actually occurs at the hinge region or just above the hinge region. Okay, and 2 fragments होता है, 2 FAB fragments वो देगा and one is your FC fragment. That means they actually fragment like this. आपका 2 FAB molecule you will get and 1 FC you will get. Okay, so that is your papin digestion. In case of pepsin digestion, pepsin actually digest करता है Ig molecule को below the hinge region. Okay, and in case of marketoethanol actually digest the disulfide bonds. It actually cleaves only the disulfide bonds. Okay, and the peptide bonds. So coming to the types of immunoglobulin molecules present in our body. See, देखो immunoglobulins 5 टाइप का होता है. ठीक है, वन है IgG, आगमेट, दैट मीन्स IgA, IgG, IgM, IgE, IgD. ठीक है, उसका नाम क्योंकि, मतलब उसका नाम क्यों है, ठीक है, मतलब why that is known as IgA, why IgG, why IgM, why E, why D. देखो, the name came due to the presence of type of the heavy chains. Okay, IgA means alpha heavy chains are there inside your IgA. In case of IgG, gamma heavy chains are there. In case of IgM, mu heavy chains are there. In case of IgE, epsilon heavy chains are there. In case of IgD, delta heavy chains are there. Okay, so coming to the salient features of different antibodies. ओके, एग्जाम में बहुत सारे क्वेश्चन यहाँ से आता है, ठीक है। देखो, फर्स्ट आईजीजी, IgG is the second most abundant antibody inside our body. Okay, and IgG is the most abundant antibody. देखो, IgG is the most abundant antibody present in our body, and IgA is the second most abundant antibody present inside our body. And in case of IgG, it is the only antibody that can cross the placenta, and it is responsible for the 3 to 6 months immune protection of the newborns. That is very very important, and it is also secreted in the placenta and breast milk. Okay, that is this is all about your IgG. Then IgM. See, IgM is the largest antibody and the first one to be synthesized in the response to an antigen. Okay, जो आपका एंटीजन आएगा उसके बाद टी सेल के साथ उसका उनका मुलाकात होगा, उसके बाद जो एंटीबॉडी प्रोड्यूस होगा ना, so that is IgM. That is the first antibody that is produced in case of any allergic or sorry allergic नहीं, in case of any antigenic response. Okay, and it is the largest antibody inside our body. Okay, and IgG, देखो, IgG is the only heat labile antibody and responsible for type 1 hypersensitivity reaction, also known as Reagan antibody. And IgD, that is the highest carbohydrate content antibody. Okay, so you can take a screenshot of this slide. This is very very important. Okay, then coming to the antigen antibody reaction. This is थोड़ा tricky है, थोड़ा conceptual है, this points are थोड़ा conceptual. Okay, क्योंकि देखो, एंटीजन और एंटीबॉडी का जब रिएक्शन होगा, that means your epitope of your antigen is going to react with the paratope of your antibody, and that is very specific reaction. Okay, that is very specific in nature. Okay, and जो रिएक्शन होगा ना एंटीजन और एंटीबॉडी, ये जो एंटीजन है ये जो एंटीबॉडी है, उनका जो रिएक्शन होगा ना ये तो स्पेसिफिक है, ठीक है। उसके बाद बहुत सारे इंटरेक्शन हो सकता है उनके अंदर, ठीक है। इंटरेक्शन मतलब जो हाइड्रोजन बॉन्ड हो सकता है, इलेक्ट्रोस्टैटिक इंटरेक्शन हो सकता है एंटीजन एंटीबॉडी के बीच में, हाइड्रोफोबिक इंटरेक्शन हो सकता है, वैंडरवाल्स फोर्सेस काम में आ जाता है, ठीक है। तो ये सब आपका लार्जर पिक्चर है, ठीक है। तो जो स्ट्रेंथ होगा ना एंटीजन एंटीबॉडी जो बाइंड होगा रिएक्ट करने के लिए, तो उनको बाइंड होना पड़ेगा। जो बाइंड होगा ना उसका जो स्ट्रेंथ है, बाइंडिंग का जो स्ट्रेंथ है, बाइंड कैसे हो रहा है, बाइंड हो रहा है ड्यू टू द प्रेजेंस ऑफ दिस बॉन्ड्स लाइक हाइड्रोजन बॉन्ड्स, इलेक्ट्रोस्टैटिक इंटरेक्शंस, वैन डर वाल्स फोर्सेस एटसेट्रा। बट जो स्ट्रेंथ हो रहा है ना, स्ट्रेंथ मतलब जो बाइंड हो रहा है एंटीजन एंटीबॉडी, उनका तो स्ट्रेंथ होगा ना, बॉन्ड का तो स्ट्रेंथ होगा ना, जो वो जो स्ट्रेंथ है ना, दो पैरामीटर है, एक होता है एफिनिटी एंड वन होता है एविडिटी, ठीक है। जो strength of the interaction between the antigen and antibody is often expressed by the terminologies like affinity and avidity. What is affinity and what is avidity? देखो, affinity होता क्या है, आपका मान लो ये एक एंटीजन है, ये एक एंटीबॉडी है, उनका एक सिंगल पॉइंट है, उनका भी एक सिंगल पॉइंट है। ये मान लो आपका सिंगल एपिटोप है, ये मान लो आपका एंटीबॉडी का सिंगल पैराटोप है। उसके अंदर जो रिएक्शन होगा, सिंगल पॉइंट में जो रिएक्शन होगा, उसको हम लोग बोलेंगे एफिनिटी। मतलब एंटीजन का भी एक पॉइंट, एंटीबॉडी का भी एक पॉइंट, सिंगल पॉइंट, ठीक है, सिंगल पॉइंट में रिएक्शन हो रहा है, दैट इज योर एफिनिटी। एविडिटी मीन्स बहुत सिंपल लैंग्वेज में समझ लो, एंटीजन का बहुत सारे पॉइंट हो सकता है, एंटीजन का बहुत सारे पॉइंट हो सकता है जहाँ पे एपिटोप इज प्रेजेंट, एंड एंटीबॉडी का भी बहुत सारे पॉइंट हो सकता है जहाँ पे पैराटोप इज प्रेजेंट। सो ऑल द पॉइंट्स कलेक्टिवली responsible for the reaction, that is your avidity. So avidity is the function of affinity, ठीक है। So avidity means the function of your affinity. Affinity means single point attraction, single point interaction, and avidity means multi-point affinities of all the binding sites when multivalent antibody reacts with a complex antigen. That is your avidity. So I hope the difference between affinity and avidity is clear to you. So coming to the types of antigen antibody reactions. Okay, so two types are there, conventional techniques and newer techniques. In case of conventional techniques, precipitation reactions are there, agglutination reactions are there, complement fixation reactions are there, neutralization reactions are there, neutralization tests are there. Okay, in case of newer techniques, ELISA, okay, Western blotting, they are there. Okay, so coming to the complement system. So what is the complement system? Complement system means complement are a group of proteins normally found in the serum in inactive form, but when they are activated, they enhance the ability of the antibodies and phagocytotic cells. Okay, so complement systems are a group of proteins. They help the antibodies and phagocytotic cells. okay, constitute about 5% of the normal serum protein and they are non-specific in nature. कोई भी एंटीजन आएगा तो वो कम में लग जाएगा. So that is complement system for you. And complement pathways, see, three pathways are there. One is your classical pathway and another is your alternative pathway and number 3 is your lectin pathway. ठीक है. What is classical pathway? Classical pathway is also known as antibody dependent pathway. Okay. Alternative pathway is also known as antibody independent pathway. In case of lectin pathway actually classical जो pathway होता है ना उनका ही एक पार्ट है, but it is antibody independent. Okay, so coming to the last portion of today's lecture, that is your major histocompatibility complex. What is it? See, देखो major histocompatibility complex होता क्या है, ये आपका जो डीएनए है ना उनका एक पार्ट है. ठीक है, डीएनए का एक पार्ट है containing a set of closely linked polymorphic genes. आपका डीएनए का अंदर में MHC is situated and it actually codes for surface proteins essential for the adaptive immune system. आपका डीएनए मान लो वहाँ पे एक पोर्शन है जिसको हम लोग बोल रहे हैं MHC. ये एक्चुअली contains a set of genes. Set of genes. MHC is actually containing a set of genes. Those genes are essential for adaptive immunity. That is MHC for you. Okay, and in case of human chromosome 6 में MHC है. ठीक है. These genes are clustered in 3 regions known as MHC region 1, MHC region 2, MHC region 3. MHC 1 and 2 help in antigen presentation to the T cell. That means जो एंटीजन आ रहा है पहले उनका मुलाकात किसी से होगा, T सेल से होगा, हेल्पर T सेल से होगा. जो ये मुलाकात हो रहा है ना एंटीजन का T सेल से, MHC 1 and MHC 2 they are going to help in the presentation of antigen to the T cell. And MHC 3 does not help in antigen presentation. Okay, but code for various proteins such as complement factors. Okay, so this is complement a major histocompatibility complex and complement system for you, and I hope this is very clear to you. Okay, so this is the end of our video. I hope you will like the video. Please like, share and subscribe our channel. Thank you.